Hello friends, how are you doing? And I hope you really all are having a great and fun time and everything is going smooth and awesome, right? So we are doing the chapter Digestion and Absorption, and in this we will talk about some of the basic questions and some important one which have the chances of appearing, right? So we'll start with the first question. What is the main function of the villi and microvilli? Before starting, let me make you clear that all the questions which we'll be doing, I'll be explaining you all of them on the basis of understanding and everything. We'll not be doing according to marks, okay, 1 mark, so we'll do one. We'll try to understand each of them in detail, and then when you're answering them, you can answer them according to the marks allotted for each of them, right? So what is the main function of the villi and the microvilli? Let us talk about this. What is the villi and the microvilli? The villi, okay, so first of all let us see what is this. The villi are foldings of the small intestine, right? The villi are foldings of the small intestine present in the jejunum and the ileum, present in the jejunum and the ileum. Right? So these are foldings of the small intestine present in the jejunum and the ileum, and what we will see further that these villi and microvilli, what they do, their main function is to increase the absorptive area, is to increase the surface area on which they will act. So what we will see that these villi, okay, and okay, before that we'll understand what are microvilli. On the villi are present tall columnar cells, right. So on the villi are present tall columnar cells with racked borders. On the villi are present tall columnar cells with racked borders, and this, okay, and this will make, right, and this will make the epithelium have a brush bordered appearance, right. The villi are foldings of the small intestine present in the jejunum and the ileum. On the villi are present the racked borders, and this will make the epithelium have a brush bordered appearance, right. So what will we see? What we will see, that say, and the villi are present, these are the outfoldings of what? These are the outfoldings or the outgrowths of the mucosa layer. So, right, here this is the villi, these are the foldings called the villi, finger like foldings, called villi, right? You know, these are the tall columnar cells, right? These are the tall columnar cells. Tall columnar cells, and this whole thing will give a brush bordered appearance. Tall columnar cells, and these are microvilli. Okay, there is a hormone secreted called the villokinin, which will increase the motility of this villi. You know, the more motile will be these villi, right? So this, you know, all together will give the brush bordered appearance. Bordered appearance, and what we see, you know, that what is the main function? The villi and the microvilli basically increasing the absorptive area or the surface area for more enzymes to act on, right? The villi and the microvilli, right, will increase the surface area for absorption. The villi and the microvilli will increase the surface area for absorption of the for the digestion and absorption of food, right? The villi and the microvilli will increase the surface area for the digestion and the absorption of the food, right? Now what will we see? Now we will see named enzymes secreted by the salivary glands of the man. Now humans have three major salivary glands, right? The parotid gland, the second one is the submandibular or submaxillary gland, submandibular or submaxillary gland, and the third one is the sublingual gland. Right? So the humans have three major salivary glands. One is the parotid gland, the other one is the submandibular or submaxillary gland, and the third one is the sublingual gland. Now the content of the saliva, we have already seen that the content of the saliva will have an enzyme, you know, the carbohydrate digesting enzyme called the salivary amylase or the ptyalin. What does it do? It acts on the starch. Okay, this enzyme acts on the starch and converts it into, you know, the further smaller products. And what are these? You know, it converts it into maltose plus isomaltose plus the limit dextrins, right? So what does it do? The saliva contains a carbohydrate digesting enzyme, right? The saliva contains a carbohydrate digesting enzyme called as salivary amylase. Salivary amylase or ptyalin. The saliva contains a carbohydrate digesting enzyme called a salivary amylase or ptyalin. Now this salivary amylase or ptyalin, this enzyme works at a pH of 6.7 to 6.8, that is an alkaline pH, works at a pH of 6.7 to 6.8. Okay, this enzyme works at a pH of 6.7 to 6.8 and in the presence and in the presence of chlorine, right. So what we see that starch in the presence of enzyme salivary amylase, right, and at a pH of 6.7 to 6.8 it will form maltose plus isomaltose plus the limit dextrins. Okay. Starch in the presence of enzyme salivary amylase 6.7 to 6.8 pH forms maltose plus isomaltose plus the limit dextrins. That is what the enzyme, hence salivary amylase. So the answer will be just a single statement. All this was basically to explain you what do we mean by that, right. Where the gastric juice is found, absolutely wonderful, where the gastric juice is found. Now the answer for this will be the stomach. The stomach has gastric glands and this gastric glands will secrete the gastric juice. The stomach has gastric glands and this will the stomach has gastric glands and this will secrete the gastric juice. So what all the gastric juice contains? Okay, the answer for my question is done. Let us talk about a little one or two sentences and then we will wrap this up, this question. So the gastric juice will contain what? The gastric juice contains, first of all, there will be the pepsinogen enzyme, right, which in the presence of HCl will form the pepsin. Now that HCl will be released by the oxyntic or the parietal cells, which we have already seen, After the pepsinogen, we will have, this will contain the HCl, right? The pepsinogen, the HCl, the prorenin, which will convert into renin, and also what we will see, that there will be the presence of, yes, mild gastric lipase. Now, why called mild? We have already stated that this enzyme works at a pH of 4 to 4.5, but here in the stomach highly acidic condition, the acidity is somewhere around 1.2 to 1.5, 1.7, okay, very acidic, that is, okay, and hence this mild gastric lipase, the activity of the gastric lipase would not be to that extent over here, right? Then what? So, the gastric juice is found in the stomach. Let us go to the next question now. What is the next question? Okay, the next question 2 marks, but as I have already told you, we will discuss the whole of the question and then probably you can decide what to write. Name the source of trypsin and the food constituent which this enzyme hydrolyzes. Absolutely right. So, see, the pancreatic juice you have already told that in the intestine will be present the pancreatic juice as well as the intestinal juice. Now, what we will see, we have already told you that the intestinal juice will act on the on the terminal, intestinal juice will be having the terminal enzymes which will break the components into the monomers. So, this trypsin will be present in the pancreatic juice. Now, this trypsin will be present in an inactive form trypsinogen by the action of the enzyme enterokinase, which is present in the intestinal juice. This trypsinogen will convert into trypsin, right? So, let us see some of the constituents of pancreatic juice, right? Some of the constituents of the pancreatic juice. The first one we can talk of is trypsinogen. The other one we can talk of is chymotrypsinogen. The third one we can talk of after trypsinogen and chymotrypsinogen is carboxypeptidase. Then pancreatic amylase and pancreatic lipase. And pancreatic amylase, right? All these are the enzymes. Wonderful. Trypsinogen, chymotrypsinogen, carboxypeptidase, pancreatic lipase, pancreatic amylase. So, we have still not reached the trypsinogen. Trypsinogen is inactive form of trypsin, and this is acted upon by the enzyme enterokinase of intestinal juice, okay, or the succus entericus, and this gets converted into trypsin. Trypsin has autocatalytic action. Once trypsin is formed, it will stimulate the other trypsinogen present to form the trypsin. Absolutely wonderful. To form the trypsin. Now, what we see that trypsin, okay, trypsin will act on protein, okay, will act on protein. So, again I will write here trypsin protein will be acted upon by trypsin of the pancreatic juice and will form the peptones and proteoses. Peptones and proteoses. Okay. The protein will be acted upon by trypsin and form peptones and proteoses, right. Hence, name the source of trypsin, pancreatic juice, and the food constituent which this enzyme hydrolyzes. So, it is protein which the trypsin will hydrolyze, and it will hydrolyze into peptones as well as the proteoses, right. Let us now move to the next question. What are micelles? Absolutely, what are micelles? So, let us understand this, what are micelles. First of all, to understand, we will see that fats, when these fats are broken, these fats are broken down into what? These fats are broken down into fatty acid, okay, fatty acids plus monoglycerides, okay, plus glycerol. This is the breakdown product of the fats, the fatty acid plus the monoglycerides plus the glycerol. Now, this is lipid soluble and it will go in through the membrane by diffusion. This is lipid soluble. However, these are not lipid soluble, these are hydrophobic, that is water resistant, hydrophobic. So, now what happens, the bile salts which are present, the bile salts which are present where? In the bile juice. And where is the bile juice secreted from? From the liver, stored in the gallbladder. Okay, the name of the enzyme called cystokinin pancreozymin, that stimulates the secretion, the contraction of the gallbladder to secrete the pancreatic juice, right. So, now what happens? So, what does the bile juice, the bile juice has? The bile juice has the bile salt, the bile acids, okay, and the bicarbonate as well too for the alkalinity. So, what happened? The bile salts has a head part and a tail part okay. So, what they will do, these fatty acids, okay, say these are the fatty acid or the monoglycerides, they will be surrounded by the what? They will be surrounded by these bile salts. Now, this is the bile salt head of bile salt, which is hydrophilic, and these are the tail of the bile salts which is hydrophobic, right. This is the tail of bile salt which is hydrophobic, right. So, now we see that this combination that is the fatty acid and the monoglyceride in the center, and all around there are bile salts, and this bile salt will have the head and the tail, and because of these hydrophilic head all around, the whole of the molecule or the whole combination becomes hydrophilic, and hence it will enter the membrane. Now, this is called as a micelle. This is called as a micelle, which now becomes hydrophilic. Later on we will see that by the process of pinocytosis, that is liquid drinking, it will enter into the intestinal epithelium or the intestinal cells, within which by the Golgi body a protein layer will be secreted around this micelle, and now it will be known as the chylomicron, and the chylomicron are they are bigger white molecules. So, a number of these molecules, when they will be absorbed into the into the intestinal villi by the lymphatic vessel, that lymphatic vessel will start looking whitish curd like appearance, and hence will be called as the lacteal, right. Okay. How would it affect the digestion of proteins and carbohydrates in the duodenum of man if a blockade in the pancreatic duct, if there is blockage in the pancreatic duct? The pancreatic duct is also called as the duct of the Wirsung, that is the functional pancreatic duct. The non-functional pancreatic duct or the accessory duct is also called as the duct of Santorini. You should all remember this. Now, so what is the pancreatic duct doing? First of all, the pancreatic duct is taking the pancreatic secretion from the pancreas to the GIT, that is to the duodenum it pours, right. We have already seen the biliary system where the hepatic duct was coming from the liver and all that, okay. I am not going to do that in detail. So, the pancreatic duct is pouring the pancreatic secretion into the GIT at the duodenum. Now, this pancreatic juice which is being poured consists of what? The pancreatic juice will be consisting of many things, we have already said the pancreatic juice consists of trypsin, isn't it? It consists of what? Trypsinogen. It consists of chymotrypsinogen. Right? It consists of the carboxypeptidases. It consists of the what? It consists of the pancreatic amylase and the pancreatic lipase. So, when the pancreatic duct is blocked, all these enzymes right, all these enzymes will not reach the duodenum of the GIT. All these enzymes won't reach the duodenum. Now, when it doesn't reach the duodenum, so these as a result what will happen, this trypsinogen, what was this? What was happening to this enzyme in the presence of enzyme enterokinase was getting converted to trypsin and was acting on protein to form peptones and proteoses. This function will not be happening, that is proteins will not be getting converted to peptones and proteoses. The chymotrypsinogen gets converted to trypsinogen and that again back to trypsin. Carboxypeptidase was breaking up the peptones and proteoses into amino acids and dipeptides. This is not happening. The pancreatic amylase will not act on the remaining starch. By the time the carbohydrates or the starch reach the stomach, we see, or the intestine, we say only 30% of the starch has been digested, 70% still remaining, but the pancreatic amylase are not there. Pancreatic lipase, most of the fats are digested here because the gastric lipase which is present in the gastric juice is not functional because of the pH over there. So, hence these enzymes won't reach the duodenum digestion and hence digestion, right, digestion of proteins and carbohydrates will be affected. Digestion of proteins and carbohydrates will be affected. Hence, digestion of proteins and pancreatic lipase will be affected. Right? Next. How is the ingested fat absorbed in the body? Absolutely wonderful. How is the ingested fat absorbed in the body? We are talking about absorption and not the digestion. The digestion was what? The conversion or the breaking down, digestion of fat converted it into monoglyceride, right, monoglyceride. So, digestion of fat converted it into monoglyceride plus fatty acid plus glycerol. Right. So, the digestion of fat converted into monoglyceride plus fatty acid plus the glycerol. Okay. Now, what we said, the glycerol is what? Lipid soluble. However, the monoglyceride and the fatty acid, okay, monoglyceride and fatty acid, these two are hydrophobic, water insoluble, right. So, what will we see? We saw that if this is the monoglyceride and fatty acid, now these will be surrounded by the bile salts, right. These will be surrounded by the bile salts, which will be having the head and the tail. This head will be hydrophilic and the tail will be hydrophobic. The tail will be hydrophobic, right. This is what? This is the bile salt. This is the bile salt. As a result, you see, because of this combination, the surrounding of the monoglyceride and the fatty acid by these bile salts, what has happened, the outside has become totally hydrophilic, that is water loving, and now it enters into, it enters how? Say if this is the microvilli of the intestine, right, it enters over here by the pinocytosis, liquid cell drinking, right. This whole combination enters into the intestinal cells. Now, once it has entered into the intestinal cell, what we will see within the intestinal cells, right, within intestinal cell, the bile salts will be removed, okay, the bile salts will be removed, the bile salts will be removed, and the whole structure gets covered by a protein layer, right, by a protein layer called as chylomicron, by a protein layer, and now this will be called as a chylomicron, okay, and now called as chylomicron. This is very important, right. So, what we see after this has entered over here, this is a protein layer, this is the monoglyceride fat or the fatty acid, and this structure is now called as the what? Chylomicron. Now, these chylomicron, as I told you, are white big particles, right. Once again, if I draw the villi, these are the villi, the folding, the folding like structures. These are the mucosal extensions of the epithelium, intestinal epithelium. Inside we are going to see what? Inside, there will be these lymphatic right. there is this lymphatic vessel. This is lymphatic vessel called lacteal, So this is the lymphatic vessel called the lacteal, where what happens, where these chylomicrons will enter. Now a lot of chylomicrons inside, will make it, the appearance of the whole thing, this vessel will look like a white milky curd, and then it is called as the lacteal, right? And hence this gets into this water, fat droplet has got into the circulation from the lymphatic vessel, it will get back into the main circulation, and this is how this ingested fat is absorbed in the body, right? What you do is you note down this, and after you've noted down, we will continue after a short session. Right friends, so I hope you must have noted down this. We will move on to the next question, right? What are the digestive fluids added to the food in the duodenum? What is their action? So what are the digestive fluids added to the food in the duodenum? In the duodenum we will see, in the duodenum, the pancreatic juice, and the bile juice is added, right? The pancreatic juice is coming from the pancreatic duct, okay, and the bile juice from, so both are coming from together by the hepatopancreatic duct, right? Hepatopancreatic duct, right? So the pancreatic juice, we have already said what is there. The pancreatic juice will have all the pancreatic enzymes, in which there will be trypsinogen, chymotrypsinogen, carboxypeptidase, pancreatic amylase, and pancreatic lipase. We have already said that. The bile juice will be having what? The bile juice will be having, the bile salts, which includes the sodium glucocolate, the sodium taurocolate, right? The glycocolate, the taurocolate, and the bile acids, right? The deoxycholic acid, the chenodeoxycholic acid, and all these, as well as the bicarbonate, which will increase the alkalinity. Also, there will be in the duodenum, the intestinal juice will also be there. The intestinal juice, right? Intestinal juice, and as I've already told you, this intestinal juice will contain all the terminal enzymes. For example, this will have the intestinal lipase, the maltase, isomaltase, right, sucrase, maltase, isomaltase, sucrase, lactase, right? Then it will be having the the aminopeptidases, the dipeptidases, right? And also, it will be having the intestinal lipase, and all this. So all this will be found in the digestive fluids, right, which are added to the food in the duodenum. We move on to the next question. How does coagulation of milk in the alimentary canal takes place? Absolutely, coagulation of milk has the protein, right? Milk has the protein. Now what we will see, as I told you already, that in the in the stomach what happens? In the stomach what happens, that in an enzyme, okay, there is an enzyme called prorenin. Okay, milk has a protein called casein. Right? Now in stomach what we will see, there is an enzyme called what? Called prorenin. Right? This prorenin in the presence of HCl will become renin. Now this renin is acting on casein, which is a milk protein, right, and convert it into paracasein, and this paracasein will combine with calcium to form calcium paracasinate. Okay, calcium paracasinate, right. We move on to the next question, that is the last question. Differentiate between the large and the small intestine. So let us draw, and not draw, let us state some differences in a tabular form between the small and the large intestine. First of all, the small intestine is longer, say about 6 meters, and this is shorter, about 1.5 meter, The small intestine has a narrow lumen, however the large intestine has a wider lumen. Most of the digestion and absorption takes place in small intestine. Most of digestion and absorption takes place here, right? However, what we see, only water and bicarbonate is absorbed in this large intestine. Only water and bicarbonate absorbed here. Okay, that's why the feces are formed, right? Now what we see, the large intestine has the presence of what? Of sac like hostra, tinea coli, and epiploic appendages. You all know what is it? Epiploic appendages. Okay, now all these three are absent over here, right? Now here there will be the folds of Kirk rings or circular pleuronaires, presence of folds of Kirk rings. However, those will be absent in these conditions, right? The small and the large intestine. In the small intestines, presence of villi and microvilli, here absence, right? So friends, these are some of the differences. We have come to the end of this session by completing all the 10 questions. Go back, revise this in the time we meet again. A very good bye from this side. Thank you.